Swayam Prabha. Digital India. Educated India.
Jnanam Paramam Dheyam. Knowledge is supreme.
Welcome to MOOC course on applications of interactomics using genomics and proteomics technologies. Today's lecture will be delivered by Dr. Mukesh Jaiswal, a senior application scientist in Illumina. Dr. Jaiswal has also extensive experience of using microarray based platforms, and he was also associated with Agilent Technologies earlier. So today he is first going to talk to you about use of microarray based platform and workflows. Particularly, you will study about antibody signatures defined by high content peptide microarray analysis. So let me welcome Dr. Jaiswal to deliver his lecture on novel technology platforms for high throughput applications.
I am going to talk about the peptide microarray. How we do fabrication on the microarray slide. Fabrication we do in the plain slide. These are simple captured by the agarose or acrylamide. Then this is the 3D slide. These are 3D slides which basically reduce the evaporation, minimize the cross contamination, and the recovery is better with these 3D slides, 3D gels. And this is the nano well. This is the micro well. So where in this well basically you can do multiplexing. So this is high definition slides basically. So these are 3 different slide format. We making the protein array or peptide array. So on these slides we do the immobilization of the proteins. One way is diffusion, another way is absorption, and then covalent bonding, and then affinity. So there is multiple ways you can basically immobilize the protein. So this is the one example where how you do the immobilization of protein by the affinity binding. The top one is the covalent bonding, right? So it make the covalent bonding on the slide and immobilize. So basically, on the glass slide, which is different type of like plain slide, or this is the 3D slide or nano wells. You basically these do the fabrication, means you can put the protein immobilized and go for then choose your samples, like it might be serum, it may be anything, and hybridize on the slide, wash it and go for scanning. So this is whole process. We make the slide in that way, right? This is protein microarray signals. So suppose if you immobilize the antibodies here, and this is the antigens, and which is labeled with different colors like red or green color. If it is specific, it's going to bind on that and give the signals, right? This is the sandwich. Basically, if the signal intensity is very less, you can intensify this by the sandwich. So basically, this is your antigen, and which we have the secondary antibody against this antigen, and it identifies the signals. Then another label free. So these are 3 different ways basically you can detect the signals. Now we are talking about the peptide microarray. How we make the peptide arrays. So how we do? So this has 24 different nodules, because 20 amino acids are there, and so we have 24 nodules, and 20 amino acid plus 4 is to deprotect basically. So how we do? So we directly synthesized this peptides on a slide. So this is 20 nodules. So basically adding amino acid added synthesized on the top and deprotected. So one by one you can synthesize 15 or 20 base pair peptide or 30 base pair peptide according to your requirement. right? And this is very important tool to analyze the epitopes. But this technology is help because you synthesize the peptide on a slide. It's not like something you synthesize outside the system and then attach on immobilize on a slide. So this is very key thing. So like standard, the standard thing. How a standard thing works basically. So pre-synthesis of peptide, synthesized before, then immobilized on the slide. So what happened? With this case, you cannot make a different type of peptides. You cannot make 60,000 peptide in your lab and then immobilize. Very difficult, right? It cannot be high throughput, high resolution, right? And then cost and customization you cannot do. So if you have this one, this Prabha print, this is our channel partner, which make this one. So what they do, like we do ATGC with base synthesis, they do the amino acid base adding on that. And they make a slide for peptide array, right? Simple addition. And this will give you high resolution, customized slide, right? So now what's the application of that? This is really important, right? What are you going to do with that? So it is used in multiple formats, multiple application. So these slides basically look like in different format. This is 2 by format. They have different formats. They have 16 by format also. It's a normal slide. So various application means they have the antibody analysis, vaccine development, biomarker discovery, autoimmune, infectious disease, and allergy. So I will cover some of that case study, right? So let's start with the antibody analysis. So antibody analysis means if you're making any antibodies to use, right? You have to know that this is going to binding in the right epitope or not. Really important. Otherwise, you cannot make the monoclonal antibody. You cannot make any polyclonal antibody, and it has to be very specific to the target, right? This is the key selling product for the Prabha print. So they have 3 different formats. So one is linear epitope mapping, conformational epitope mapping, and then substitution. So there are 3 different ways they make a peptide array, right? So let's start with linear, right? So suppose this is the antigen sequence, right? This is the antigen sequence. So what they do, they make an overlapping peptide of this antigen's sequence. So these are the antigen sequence, and they are overlapping peptides. So they make overlaps in that, and this overlap peptide is basically synthesized on this peptide array. You got this antigen sequence, you know that, but you doesn't know what's the epitope for that, right? So then they use the target antibody, which you want to define the epitopes. Secondary antibody, then this is going to scan, and the signals basically where this base is detecting, that's your epitope. So this is the epitope where the antibody positively bind. So this is the monoclonal is only for this epitope. If it is polyclonal, it's going to bind several places, right? So it's very easy. How did they define the linear epitope? So just the overlapping peptide they use, right? So if you use the linear one, you basically see the epitopes difference very clearly. Here in the standard we cannot see. Resolution is not that great. So now the conformational one. So if sometime some proteins are basically like CD20, this is the extracellular, this is the transmembrane protein, and they have the extracellular moieties like this here, and these are the epitopes, right? This one, this part. So, but this is not linear, so it's very difficult to identify the linear conformation. When the conformation is there, you cannot use that strategy for the detection of epitope. So what they do, they make a very similar type of structure like 7, 11, and 12 conformational peptide, circulated, same conformation, right? And imprint on a slide. So this is circular one. Similarly, they look in a protein form in extracellular. They make in a slide basically, like this format. And now when they use this conformation and this conformation, right? See this, they're getting the positive signal from this for these epitopes. But if it is linear, you don't see any signals, right? So they're clearly showing that there are 3 different epitopes, amino acid of 7, 10, and 13, right? Because they make the conformation for that way where the antibody going to bind, right? So this is the another way. If the protein are like some conformation part, you can make the peptide for this one. Because linear doesn't not going to work. Because if it is linear, the antibody is not going to bind. It's something conformation thing, right? Then this is the substitution one. So substitution means this is another way. This is again, this is the CD20 protein, and they're testing their targeted antibody. So they are thinking that suppose this is the epitope for that, right? This is the antigen sequence for that. What they do, they replace each amino acid by 20 amino acid, right? See, means alanine is replaced by the 20 different types, right? So each amino acid of that sequence is replaced by 20 different types. You got it? That's a substitution. So when they replace, so see, this is the cluster of the things where the antibody binds, but there are many of the amino acids they're going to bind. So clearly this is not the clear epitopes, right? But see this cluster, this is the cluster where only there's no signal, right? That means if you replace that where antibodies bind and they are not getting signal, that means they are the clear epitopes because the amino acid replaced. So these are the epitopes where they are not getting any signal because of the replacement of that amino acid, right? So these are the clear epitopes. These are the epitopes basically where they do not get any signal by the replacement, right? So this is the 3 different ways basically the peptide is used for the conformation, means for the epitope study. Any antibody synthesis, you need to check for the epitope study. You can do easily the protein peptide array. So coming to one example, let's start with an example with the autoimmune epitopes. This is the epitope microarray. This is the freely available in NIH website. This sequence are freely available, and that is basically used for the autoimmune study, right? So it has 4,287 linear autoimmune peptides on that. So you can detect various number of autoimmune disease. So this is the example. So what they do, they took the patient serum of 56-year age of diabetes mellitus type 1 patient, and they have it as on a slide to check the autoimmunes, right? And see they getting, so the green one, this one is the IgM signal and this is the IgG signal, response of the antibodies, right? So this is the IgG, this is the IgM, and if you see this is the protein tyrosine phosphatase, this is the regular, if you diabetes mellitus 1, this is going to get this signal. So the very positive signal for protein tyrosine phosphatase. And there are multiple signals you're seeing basically for some different type of viruses, right? But you see the signal for the IgG response is very high. That means the autoimmune response in this patient is very, very high signals. So that type of study you can basically do with these array and analyze what type of different biomarkers for the autoimmune disease also. So I'm concluding with it can be used for the different diagnostic, and you can use this one for the high throughput scanning of the serum, multiplexing, and basically the overall idea, try to develop their own arrays and test for the population. We're really working for some infection disease or autoimmunes, right? And this will definitely help for the biomarker studies.
Well, actually you may have realized that there are a variety of microarray platforms which can be used for different applications. In fact, vendors make different type of printing system, different type of scanners which could be used for your applications. You can choose rightly that which kind of instrument and technology is best suited for your applications. In this case, they have used simply their own technology to scan the slides using two color systems. Now, this gives you the good flavor of technologies used for microarray based workflows. From next lecture onwards, you will learn about different techniques for studying biomolecular interactions, and I am sure you will be really excited to move forward, that now you have identified a potential protein of interest which looks a potential candidate biomarker or a therapeutic target. Now, which are the possible interactors which could interact with this protein of interest, and which are the technologies which can be used to study this problem. So, we will continue this discussion in next lecture. Thank you.